Hello and welcome to lecture 16 of our course on stochastic modeling of biological processes. In this lecture, we will start our discussion of chapter 6, which is about stochastic modeling of reaction-diffusion processes. To motivate this chapter, I'm starting here with the deterministic modeling of reaction-diffusion processes, which students taking this course in Oxford can learn in other mathematical biological courses we offer to our undergraduates. Deterministic models are written in terms of partial differential equations for spatially varying concentrations of chemical species. Such a reaction diffusion equation is written here, where Ci is the concentration of the ith chemical species and Di is its diffusion constant. Students who took courses on deterministic modeling of biological processes will know that such reaction-diffusion equations can generate spatio-temporal patterns, like Turing patterns. In this video here, I am illustrating a simple pattern-forming model from the developmental biology. A diffusing morphogene is produced on the left part of the domain and degraded everywhere. Such a process creates a morphogene gradient, which can be interpreted by cells. Here is a layer of cells. A cell can have three different states depending on the morphogene concentration, and we obtain this French flag model, which is in the lecture notes as figure 6.7. Reaction diffusion partial differential equations include two types of terms on their right-hand sides. The blue term corresponds to reactions. It is a function of the concentrations of chemical species. Considering chemical reactions on their own, the deterministic model is written for concentrations which are only functions of time, as the following ordinary differential equation system. The corresponding stochastic modeling was covered in chapter 1, where we discussed the Gillespie algorithm and its analysis using the chemical master equation. The red term corresponds to diffusion, and we have discussed the corresponding stochastic models of diffusion in chapter 4, including molecular-based modeling in section 4.1 and compartment-based modeling in section 4.2. In this lecture, we will use the compartment-based modeling of diffusion, and we add reactions to this modeling approach, presenting compartment-based stochastic reaction diffusion models. In the following lecture, we will discuss molecular-based approaches to stochastic reaction diffusion modeling. This is a research area with a lot of beautiful videos and visualizations available. Last year, when my lectures were delivered in person, I played some videos of stochastic reaction diffusion simulations of real biological systems in my lectures. Since my lectures are online this year, it could be more valuable for you to pause this video and simply Google some of these software tools to get an idea what type of software packages are available. These software tools have been developed by computational biologists. Chapter 6 of the lecture notes provides mathematical analysis of the underlying stochastic reaction diffusion algorithms. So we will use simple illustrative examples, and we will not use any software packages. However, for simulations of real, complex biological systems, it is useful to use the available software packages whenever it is less straightforward to write your own codes. For example, when the system under study includes a very complicated geometry. In lecture 16, we will discuss a compartment-based reaction diffusion algorithm, which is used in software packages URDME, Lattice Microbes, MesoRD, Smart Cell Steps, etc. Molecular-based approaches to stochastic reaction diffusion modeling are used in software packages called Smalldyne, MCell, Green's function, reaction dynamics, and many others. In section 6.1, we use an effectively one-dimensional problem. We consider an elongated domain, a cuboid of length L and side H. We assume that molecules of chemical species A diffuse and are degraded in the whole domain. Molecules are produced in the red part of the domain, and there are no molecules in the system initially. To implement this model in the compartment-based stochastic reaction diffusion approach, we divide the computational domain into k compartments, and we denote by AI the number of molecules in the ith compartment. Then diffusion is described by this chain of chemical reactions, which we studied in chapter 4. Degradation happens in every compartment, and the chemical species A is produced in the red compartments. Therefore, this reaction diffusion model is given as this system of chemical reactions, which we can simulate using the Gillespie stochastic simulation algorithm with the results presented in this video. The black line is the solution of this partial differential equation for the concentration of molecules of chemical species A. The first term on the right-hand side models diffusion, the next one describes the localized production, and the last one is the description of degradation. We use zero flux boundary conditions, and the initial condition is A is equal to 0. We observe that the molecules are produced in the left part of the domain, they diffuse and are degraded everywhere. Since this model is written as the first order chemical reaction system, we can analyze the corresponding chemical master equation using the techniques which we presented in chapter 1. The mathematical analysis of this model is left to you as exercise 6.1, where you will show that the equations for mean are equivalent to the discretized version of this partial differential equation. So it is not so surprising that it seems to describe well the average behavior of the system in my illustrative video. Snapshots from this video are presented in figure 6.1, showing the numbers of molecules in compartments at times 10 minutes and 30 minutes. Another compartment-based stochastic reaction diffusion model is presented in section 6.3. This model illustrates that it is relatively straightforward to implement any reaction diffusion system using the compartment-based approach. In section 6.3, we consider a system of two chemical species called A and B, which diffuse in the same elongated domain as in our previous example. They are subject to 6 chemical reactions. 5 of these reactions happen in the whole domain. They include 2 second-order reactions. A reacts with A, and the product is not further considered. A reacts with B, and the product is not further considered. So these 2 reactions are removing molecules of A and B from the system. Then we have the degradation reactions of both A and B, which again remove molecules of A and B from the system. Finally, we have the production of A and the localized production of B. We assume that B is produced in this red part of the domain. To write the compartment-based approach, we again divide our computational domain into compartments, and we model diffusion of A and B as 2 chains of chemical reactions, one for A and one for B. The first 5 reactions will happen in all compartments, and the last one only takes place in the red compartments. In summary, the compartment-based model will again be written as a large system of chemical reactions, which can be simulated using the Gillespie algorithm. As an exercise, you could write this chemical system down yourself. If you have any difficulties with this, you can find the resulting system on page 167 of the lecture notes, where we also compare it with the corresponding deterministic model, given by this system of 2 partial differential equations for concentrations of chemical species A and B, which is solved with zero flux boundary conditions. The results of an illustrative realization of the Gillespie algorithm applied to the compartment-based reaction diffusion system are presented in Figure 6.3, where we plot a snapshot of both A and B. Since B is produced in the right part of the domain, B is an increasing function of the position. Although A is not involved in any localized reaction, we still obtain a non-constant solution for A, because A reacts with B. So if we have more B in this part of the domain, then we will have less A in this part of the domain. The black line is the solution of the deterministic partial differential equations. Since this system contains second-order reactions, we know that the black line will not exactly describe the average behavior of the stochastic model, but it provides a reasonable approximation of it. So, to summarize sections 6.1 and 6.3, we have showed that compartment-based reaction diffusion modeling can be implemented by dividing the domain of interest into small compartments and modeling diffusion as jumps between neighboring compartments, as we did in Chapter 4. We then assume that chemical reactions can only happen between molecules in the same compartment. Small compartments are assumed to be well mixed, and we write a compartment-based model as a large system of chemical reactions. So we can use the Gillespie algorithm or its equivalent formulations to simulate the reaction diffusion systems. Since the resulting chemical system can be relatively large, its stochastic simulations can be computationally intensive. In the lecture notes, you can also find Chapter 5, which is not covered in our videos, and which discusses various methods for improving the efficiency of simulations. I will conclude this lecture with a discussion of section 6.8, which is on pattern formation, discussing how we can go from a layer of cells to a pattern. The first model is the French flag model, which I have already illustrated in the video at the beginning of this lecture. In this model, diffusing morphogene is produced in the left part of the domain and degraded everywhere, which is exactly the model studied in section 6.1. Here I have visualized the same results which I calculated for this model using two thresholds. If the number of molecules is less than 30, then the red program is initiated by cells. Values between 30 and 80 molecules correspond to the white program, and the values above 80 correspond to the blue program. With this interpretation, I obtained the video of the stochastic French flag model here. Stochastic modeling gives us additional details, like the description of the size of stochastic fluctuations, variance of the number of molecules, which we can obtain from this model both computationally and analytically. The second patterning model is included for those who have previously took a course on deterministic modeling of biological systems. It is the stochastic version of Turing patterns. Our illustrative model is written in terms of two chemical species, A and B, which are subject to four chemical reactions. The corresponding deterministic model is a system of two partial differential equations for concentrations of chemical species A and B. Under some conditions on the parameters, the homogeneous solution is unstable and spatial patterns will develop, which I have again visualized here using thresholds. The left panel shows the snapshot of A and the right panel shows the snapshot of B. Large values of A are visualized using green color and the lower values of A are visualized using black color to illustrate the resulting pattern. This brings me to the end of lecture 16, where we have discussed compartment-based modeling of reaction diffusion processes, covering sections 6.1, 6.3, and 6.8. Please read the corresponding pages of the lecture notes before watching the video of lecture 17. In our next lecture, we will discuss molecule-based approaches to stochastic reaction diffusion modeling. Thank you very much for listening to lecture 16 of our course on stochastic modeling of biological processes. Bye bye.